gene therapies. We will start with Professor Kathrin Meyer from the Ohio State University and Nationwide Children's Hospital Research Institute in Columbus, Ohio. Kathrin has been trained in Switzerland at the University of Bern, and for several years now has been doing excellent work at the Nationwide Children's Hospital in Ohio, developing gene therapies for neuromuscular disorders. She was involved in the development of the gene therapy for spinal muscular atrophy that we now have in the clinic for the patients. And she works also on several other models on inherited neuropathies and neurofibromatosis, among others, as well as neurodevelopmental and autism spectrum disorders. Kathrin really has the complete experience all the way from the preclinical to the clinical translation of gene therapies. So she will really give us an overview, and especially the lessons learned that can be transferred to any gene therapy development for the disorders of interest to the ERNs. So with this, I will give you the podium, Kathrin, and also I would just like to note that we have two talks today. After Kathrin's talk, we will go directly to the second talk by Marta Kollb-Sielecka, and then we will have the question and answer session at the end. So thank you again, and the floor is yours, Kathrin. Thanks.
Yeah, thank you. Thank you, Kleopas. Very nice to be here today and talk a little bit about the work we've done and share some thoughts really about it as well. All right, let me see. These are my disclosures. So I just want to say upfront that I'm a huge fan of gene therapy. It might not sound like it as we're going through the slides because I'm talking about challenges, but I do think it is an incredible tool and it does have huge potential. There are some things we have to figure out, but I like to put that statement in the beginning so you can so you don't think that I'm here to talk about all the problems only. Quickly, our center, if you haven't heard about it, Dr. Mendel, who just retired, really was spearheading the development of the center together with Phil Johnson, neuromuscular diseases, children and adults, and neurodegenerative diseases. And I think it's 15 or 16 AAVs that we have brought to the clinic in the last years. And also there's a bunch of companies that have been built around these technologies. We also, I believe, are the only institute that has two FDA-approved, fully approved drugs on the market at this point. Of course, not just us; these are all company collaborations. But just seeing, at least pieces and parts of what it takes to get it not just to the clinic, but also to the approval and actually to the market, it was really interesting from my point of view. I'm not going to go into a basic gene therapy education here, but I like to put this on just to remind everybody what gene therapy actually means. And I do think, we are still at this point trying to treat mainly monogenetic diseases where we have a better understanding of the disease mechanisms. But then also, I think, unfortunately, the term gene replacement that is currently used, I think, by a lot of researchers, it doesn't really make any sense because it doesn't stand for gene per se. It only stands for the coding sequence, so the protein that's made from a certain gene. And it doesn't stand for replacement either, because the way we're using gene replacement, we're thinking about classic AAV gene therapy, which is basically just bringing in a working copy that allows to make a healthy protein. And I want you to keep that in mind maybe for some of the second part of my talk, when I talk about other strategies that we can use to potentially leverage the entire gene, if one copy is intact. Gene replacement could potentially be true if you are swapping out the genomic area where your gene is in, but that's a whole level of complicated for neuromuscular diseases or neurological diseases. Okay, this doesn't want to move. All right. This is a slide I've been showing for a few years, but I think it still holds very much true. A lot of the gene therapy trials are still in these very early phases, phase 1, phase 1, 2. Only a few of them actually have made it to the later stages, phase 2, phase 3, and BLA-enabling studies. So we are still learning and trying to understand a lot about these therapies. Maybe not, we're not in the infancies, but I would say maybe in the teenage years at this point. And I think some of the issues that we're having, which I guess any drug has, is that we are using models that don't necessarily reflect what's happening in the clinic. Especially mice, which are monogenetic, they are all very similar. And that's absolutely not true when you think about patient populations, which are very diverse. We can use large animals to bridge some of those gaps to understand dosing better. So you don't have to go from an animal that's, you know, as big as your fingertip to a human being, but there are still some additional challenges there, which I'll come back to on the next slide. So really what we need to do, and that is a hard thing to talk about sometimes, especially if you work with patient foundations, is we have to learn from the clinical trials. We might not get it right the first time around. We're trying to do that, but sometimes, unfortunately, that is not the case. So there are translational gaps that are very difficult to overcome between the model systems that we're working with, the mice, large animals, and the patient population. Certainly size is one. But even if you forget about the size, the differences in how the nervous system, for example, is built, how many glial cells you have versus neurons, et cetera, the ratio of brain to spinal cord, all of these things are different. And then in the mice, in most cases, we work with knockouts. So the gene is completely ablated, or you ablate protein production completely, versus talking about patients with different mutations, different point mutations that might have residual functions or could interact with the healthy protein that you are trying to express. The other aspect that I think is oftentimes forgotten is that these animals live in very standardized environment. They eat the same thing, they sleep at the same time, they have the same exercise opportunities and the same people taking care of them. And when you go to the larger animals, in most cases, we use wild types because it's very expensive to make a large animal model. And then it also takes a lot longer oftentimes until you see a disease phenotype. So really, it's not very suited for disease modeling. But even in the wild type animals, you still have the same nutrition, the same exercise paradigm, the same lights off at 6 o'clock in the evening, very standardized living conditions. Patient population, not so much. You know, every family is different. Every family takes care of their children differently. They might sleep at a later time. A lot of patients have different standard of care. They are taking medications. We don't know if those medications have any impact on transduction or expression of the genes that we are interested in. So very, very variable, extremely variable, and not even thinking about the genetic variability, just the lifestyle and nutrition, et cetera, et cetera. Sorry, this keeps getting stuck up. So another thing that we really struggle with, which is different from the preclinical work, is that the patients are treated at different time points in their disease progression. It's very hard to standardize your patient population as close as you can do it in the mouse, right? And so the experience that we have from multiple clinical trials across the board, not just at Nationwide, is that earlier treatment is better. It's a lot easier to prevent damage than to restore damage that has already occurred. But if you have if you work with a disease that is mild and you treat the patient early, then you have a very long clinical trial, and that is expensive. And unfortunately, it's really hard to justify that towards, for example, investors and other sources of finances, because they are expecting an answer in a year or 2 years, which is literally in many cases impossible. So which patients do you select? Do you select the more severe patients, hoping to get a fast answer, but you might not know if they can respond? The treatment window where you really can have efficacy might be a lot shorter there. So all of these things really complicate the clinical trial design. And excuse me, I think part of these complications are responsible for, I would say, some of the disillusionment, some of the frustrations or news that came out where clinical trials failed their endpoints. Probably not the only explanation, but after the successes of Luxturna and Zolgensma, and in a very enabling economic situation, there was a real excitement for the AAV gene therapies. And at least from where I sit, we've come past that excitement. Some of the things we thought were going to work didn't work the way we wanted. There were unexpected complications, et cetera, and we went through this disillusionment. But I think we also did learn a lot of lessons in that process, and hopefully are now on our path to enlightenment. I hope to think so. So why are we doing this? It sounds like it's really complicated. So as a PhD student, maybe I'm like, maybe I'll stay away from this. This is really complicated. But I think in most cases, if we select diseases where really there are no other treatment options, or there's very substantial room for improvement, there's a lot of potential for these therapies. The viral gene therapies are the most efficient in terms of spreading the therapy across really big organs, especially the nervous system, where a lot of drugs don't make it to. Oftentimes, these are severely debilitating disorders. The patients are really, really sick. And especially the viruses are good to get to broad biodistribution. And then also the fact that potentially with a single treatment, you can have a life-changing, long-lasting effect. We don't know how long, but for now, many of these patients are 7, 8 years past treatment and they're still doing really well. As I mentioned earlier, the redosing is extremely challenging because once you dose the patients, you basically immunize them against the treatment. And it's really difficult to find immunosuppressing regimens that are strong enough yet safe in a patient population that's already sick to be able to redose. So we're trying to get this right the first time, and that is really challenging. I mentioned this, especially for the nervous system. I think these are really good therapies. So one thing that I've spent a lot of time on is trying to find ways to reduce the toxicities that have been seen in clinic with the intravenous delivery. There have been several deaths in AAV gene therapy trials, as you might have heard, that are oftentimes related to liver toxicity at high doses, especially when the drug is delivered intravenously. There were some issues also with kidney and heart that unfortunately led to deaths in these trials. The spinal fluid delivery has been a lot safer, and we know from our data and other data that with the exact same virus, you can use at least 10-fold lower doses, maybe more, to have the same effect. This is an example from our team. ALS is a very severe disease. The untreated mice die at around 130 days. And the green and the blue curve are mice that were either treated intravenously or via the spinal fluid. And as you can see, we have the same result, but with a 10-fold difference in vector concentration. So one thing that we learned is that you can extrapolate doses between different programs if the cellular localization of the proteins are similar. So for intracellular proteins, it seems to hold true that the more cells you can target safely, the better you're going to have, the better efficacy you're going to have. This might be different if your protein of interest is secreted, but at least if it is an intracellular location, you can really learn and leverage the experience from previous programs, which can help reduce the number of experiments that you have to run to some extent. One thing that I'm really also starting to think about a little bit more is, even if you have patients that, at least from the outside, look similar in terms of disease progression and time of treatment, you can see variable responses. Some patients have an amazing response and some patients don't. And unfortunately, right now, we don't understand why. We can't really discern which patients will have a fantastic response and which not. And that is something that I think we should put more effort in. Of course, one part could be the immune system. Each patient has a different immune system, all the way from the native immune system to the adaptive immune system. Those could play roles in terms of how much virus actually makes it to the cells. There can be differences in CSF leakage or even in blood circulation between the patients. And one thing that we are looking at also is, are there differences in the metabolism, and do differences in the metabolism change the uptake of the virus or the efficacy in which you get expression of your protein? And this is unpublished data from my team where we use the exact same virus at a low dose so that we can get a comparison. The virus encodes for green fluorescent protein. And as you can see, just by manipulating the metabolism of the mice, we can change the level of expression of these proteins, which coincides with a higher uptake of AAV. This is something that I don't think we necessarily look at enough, and it's really important in the clinical trials, if you can, to verify whether your AAV actually made it to your target. It's a little bit complicated if you work with the nervous system, but if you work with the muscle or the liver or maybe some other peripheral organs, you have a better chance to do that. And also, again, selecting the patients as homogeneous as possible, but also then really trying to collect additional information that could then help you maybe pass clinical trial to evaluate the differences between the patients as well. This is not just something that my group is exploring. This is an example that has been published by a Dr. Park's group, where they looked at AAV2 and AAV9 uptake in the retina in mice that had diabetes and mice that were healthy. And you can clearly see, again, there is a big difference. So this could be problematic potentially because we never study in our dose response curves. At least it's not very common to study the uptake or the expression, the effect of your virus in the context of different metabolic factors that you will maybe see in clinic. Another issue that we often see is that a lot of the AAV vectors have tropism differences between species. Some work really well in mice, some work really well in humans, and anything in between, and you can't expect that it is the same throughout the species. So you might need a higher dose in a mouse than you need in a larger animal to see toxicity or efficacy. So large animal studies are certainly very important, but the large animals are not sick. So then if a patient has, pre-existing liver conditions, for example, that patient might be weaker compared to the large animal that you did your safety study in. So these kind of aspects, oftentimes we have to be very careful in the selection of the patients, or unfortunately start with doses that we hope to be efficacious, but maybe not the maximum efficacious dose that we would like to give all the patients. And this is something that I think is also worrisome a little bit with the new capsids. I think all the new capsids that are currently developed it's a very exciting field, but we don't know how they behave in clinic. At least with the capsids we have now, we do know the devils we're dealing with, and there are some protocols that have been developed to combat the side effects that are seen. So with the new capsids, that will be a whole new ballgame that we have to get through. Last, it's already 20 minutes, so I'm only going to show you a few more slides. even though we are able to deliver these AAVs via the bloodstream or via the CSF pretty efficiently to the brain, the biodistribution isn't even. It looks a little bit like the skyline of Manhattan. There's ups and downs. There is areas that are targeted really high. There is areas that are barely targeted. And that is so the doses that we use now are limited, unfortunately, because of the tissues, areas that are targeted at a really high level. So we cannot get more to the deeper brain regions, for example. So this is something we also have to figure out. If we had a more even biodistribution, we could probably more safely increase the dose, or maybe we wouldn't even have to, and the efficacy would be better as a consequence. Some of it can be done with modeling. This is one example from a company that I am working with, but there is other companies and other teams that are working on trying to improve the delivery to the nervous system to make it more homogeneous and also understand how the data from the monkey actually translates to the patients. Because again, a monkey is not just a little human. The ratios of brain to spinal cord and the spinal fluid flow are slightly different, and we are trying to optimize for dosing in the patients. I think one of the things also for the preclinical researchers, and I think I'm going to stop after this, but this is a very important message. It's very hard to compare the data that's out there from the publications. There's a lot of different ways to inject your AAV. There's and most of the publications, of course, are big fans of what they did. They say, oh, this method works really well. But it depends on, or we can use lower amounts of AAV than what was previously published if we use this method. The problem is you have to really use the same AAV prep if you want to make these comparisons, because if you don't use the same measurements to determine the concentration of your virus, all of those comparisons are kind of mute. And it's not just the location of the injection that will change your biodistribution. The volume of the injection, the speed of the injection, what you mix your vector in, all of these things can change how your drug moves through the nervous system, moves through the CSF. And so some of the statements that we see in the manuscripts, of course, they are just a lot more general than this. So be careful when you compare different publications when you're trying to select your mode of delivery. It might be better if you do a side-by-side comparison yourself. That will give you probably a lot more information on that end. I'm going to stop here. I had a lot more that I wanted to tell you about, but maybe we'll do that next time. So I just want to say thank you so much for having me, and I'm looking forward to the question and answer session.
Thank you very much, Kathrin. This was really a very insightful presentation, and we would love to hear more, of course, but we have also the question and answer at the end for anything that is not clear. I'm sure there are a lot of issues still to discuss. So thank you very much. We will go now to our second speaker today. Is Marta Kollb-Sielecka from the European Medicines Agency in the Netherlands. She will talk about the regulatory approach to evaluating and approving gene therapies for neurological and other rare disorders. So this is really very relevant for our mission and for our ERNs. Marta Kollb-Sielecka joined the EMA in 2013. She is a product lead in the Therapies for Neurological and Psychiatric Disorders Office, as well as a member of Advanced Therapies Medicinal Products Matrix team. She holds a master's degree in pharmacy from Warsaw Medical University. Prior to joining the agency, she held a number of positions in pharmaceutical industry. So thank you again, Marta, for accepting to present this very important topic. And please, the floor is yours, and we're eager to hear your presentation. Thank you.
I'm sorry for lack of video. My connection wasn't very stable, but the audio will hold. So you see the ATMP in this title because in the EU framework, gene therapies fall under the wider ATMP umbrella. So my talk will be generally about ATMPs, but also about the gene therapies in particular. So the disclaimer as usual, and I have no interest to declare. So I will talk about the EMA role and how you fit in the wider network, what are the ATMPs and why they need their own framework, what is the pathway for approval, and what we can do to support the companies and academia on their journey. So EMA is an agency of the EU established in 1995, first in London, then moved to Amsterdam. After Brexit, this is our new building, which we only got just before COVID. So you often hear in the media that EMA approved something or refused something, but the truth is we are just a part of existing EU-wide network. So we are more responsible for the coordination. Whilst we really are drawing on expertise from all the member states. So we have around 900 people working at the EMA, but we draw on thousands of assessors and experts across all member states. And they are the people who form our committees, the working parties working on the guidelines. They create teams to conduct evaluations of all the applications EMA receives. And we have already issued over 1,000 of those authorizations over the past years. So I mentioned that the gene therapies in Europe fall under the ATMP umbrella. So they also include the cell therapies and tissue therapies. And gene therapies, obviously, our focus here. I won't go through the definitions. Everyone knows them. But okay, this is, you can see that from the ATMPs approved in the past few years, gene therapies form the biggest group, and that's both the in vivo and ex vivo modified genes. And also you can see that the density of the graph increases as the timeline goes forward. So I'm more optimistic. I think that we have more also on our radar. So I sincerely hope that this will be an active field going forward. So this is a comparison of traditional versus ATMPs medications, because all of those aspects do trigger certain regulatory actions. So ATMPs, as we know, they are usually given, and gene therapies are given in a single or few administrations, but the effects are supposed to last ideally lifetime. They usually require a specialized centers for treatment, which means that those centers have to be qualified. Our label would usually also include very precise training materials, qualifications for the team. They're also difficult to copy. We have generics and biosimilars for the other molecules. This is not the case for ATMPs or gene therapies. And also in the approval, we think of the process, not just the drug. As we heard before, they have to be given often via surgery. So the patients have to be preconditioned. So the efficacy and safety of this is taken into account. And again, we cannot stop treatments for non-responders. So the benefit risk decisions have to be taken very carefully. The last two items on that list is a cost and market access, and those issues we are aware of, but these will never be part of regulatory decision making. So this is something which is happening post-approval. And that's the entry route of gene therapies in the EU. Once all the clinical trials are conducted and we have data and the quality, the manufacturing is done, we have a dossier, which is sent to us at the EMA, and it's taken mostly by a so-called CAT, Committee for Advanced Therapies. And this is a committee specifically created with expertise to evaluate ATMPs and gene therapies. They will have a few rounds of assessment, giving applicant a chance to respond to questions. And that process takes around a year, can be much longer if the questions are difficult, if there are some gaps in the dossier. So I have seen some of them, some of the evaluations can be after the CAT and CHMP make the decision, we forward this to the European Commission for their formal license decision, and then this goes onto the market to the payers and HTA negotiations. But you can see this loop on the slide that after launch, the EMA still keeps eye on the medicine. We always have, for gene therapies, very well-designed program of follow-up, registries, other studies, real-world evidence. All of this data, all of this data will find its way back to EMA, to CAT, other committees. So the label can be fine-tuned. We can add warning, remove warning, fine-tune indications even. So there is a very, very intense post-approval activities as well. Things we take into account, the quality aspects. We have seen that upscaling production from the clinical trial batches to commercial ones presents many problems. So, the GMP certifications, need for inspections can also delay this approval process. As I mentioned earlier, the surgery, the process of administration can have efficacy and safety impact. You have to assess it very carefully. And it could be difficult to disentangle the safety of surgery versus the safety of actual administration of the gene therapy sometimes. Patients preconditioning, steroid regimens, those are also discussed in details. We have in our labels very precise information of for how long they should be given, what doses, what should be the liver enzymes control levels, and so on and so on. The other gene therapy specific items, there's obviously a risk of target insertion, and that can create problems down the line, hence long-term follow-up mechanisms. And again, shedding for gene therapies could be, even if the risks are very small, we still have to conduct the risk assessment for the environment. Is the risk to the caregivers? So it's usually not a problem, but it still have to be conducted. And go back to clinical development aspects, which already have heard earlier from Kathrin. The trial design is often a problem. The dose finding is not as it was done in the old-fashioned development, when you can have more patients and easier dose finding designs of trials. Because gene therapies often target rare diseases or even ultra rare, we do have single arm trials, but then it's really important to design your control arm well. So if it's historical controls, natural history studies, they have to be done very well and very carefully, because for some conditions, the data is of good quality, for some, sadly not. So I have to really urge you to think about control arms very carefully. Indication considerations. Sometimes it's very simple. The indication will be almost the same as the population in the clinical trials, but it may have to be restricted based on efficacy or safety. It could depend on the patient's age or stage of the disease, or even weight. And again, extrapolation to possibly wider indications can be possible, but then we have to be also very careful on which grounds this is done. Safety data is also quite limited at the time of approval because of the number of patients. So that's why we have the long-term follow-ups planned already. Similarly, with the efficacy, we don't know yet if the response will be a year, 5 years, 10 years, lifetime. So we always encourage applicants to start planning early, registries, phase 4 trials, any other forms of follow-up to be able to bridge the gap between what we have at the time of approval and what we actually need to know. And this is also a plea to all of you to come to us early, because EMA has a wide number of processes and procedures to aid the developers as early as possible, so advices, innovation task force, etc., etc. I also want to mention PRIME process. PRIME is the early access program in which very promising molecules can be given special support from the EMA, and also it looks obviously good for the press release if you are assigned this by the agency. And if this works, I hope it will. You can also think about website. We have a kind of here checklists and flow charts. So if you wonder about how to develop in as well, and for instance, the flow chart for non-clinical development, which gives you also links to the guidelines. Also, one other aspect I want to highlight here is that we don't only work with industry, with companies, we also want to encourage academia and non-profit organizations. So there's a currently ongoing pilot in which we provide enhanced support to some applicants from this sector, and we can guide them through the planning and submission of their applications. That also helps us to identify any gaps we have in the current tools and the guidance, so we can also improve our guidance based on their experience. So I think the key message is here that we have a fairly well-established legislation and framework for ATMPs and gene therapies, and many already approved. But we have to also warn you that the evaluations of those are subject to the same molecule. So you have to demonstrate a standard of quality, efficacy, and safety to be able to get approval in the EU. And again, because of the usual limitations because it's often orphan conditions, rare diseases, you have to get the data right very early on, and you have to plan for the long-term collection as well. All of those products, they very often push boundaries. So we have to also engage earlier to understand each other. And hopefully that would smooth the process when it comes to approval time. So that's it from me. This is the questions time, and this is my email for the future as well.
Thank you very much, Marta. This was a very insightful and useful update on the whole process of regulatory approvals for gene therapies. Maybe I can start before we go to the questions. I can start with a question that I have. So I wasn't sure if it was clear, but do you have a process similar to pre-investigational drug evaluation that FDA has? So evaluating early on at the preclinical stage, the development process, the data from the preclinical studies, and maybe providing input on the design of clinical trials before they start?
Yes and no. So I think the FDA has this whole IND program, which is they have much more oversight of development. We do it differently. So the applicants have to approach us with questions. So we don't do it for everyone. It's more of an applicant's initiative that they have to come to us with defined questions, and then we can answer them. There are other ways. There's more options for the ATMPs from the small and medium companies. There's something called ATMP certification, and that's when you are towards the end of preclinical, early clinical. You can come to us for some kind of checks like that. But sadly, there's no IND-like system. We have sometimes dossiers which have been developed entirely out of Europe, in Asia or US, and we see them for the first time at the MAA stage. So that's obviously then slows the evaluation somehow, because we as network have to understand this from zero. Whilst if you come to us for advice earlier, we can engage better.
Okay, so although there is no official process similar to pre-IND and IND, probably there is a way to communicate and try to engage early, if possible.
Yes, yeah. I think, there's obviously, you have to come to us with the pediatric investigational plan. So you have to do that to be able to get approval. You have to come to us separately if you are developing an orphan product for the orphan designation. So I think there are multiple tools. So the more of them you use, the better.
Great. Thank you very much. We will look at some of the questions now. So the first one, the first question is based on what Dr. Meyer is saying, the N of 1 studies should be much easier. Is that right? So I think this relates to the variation between patients in trials and the challenges related to what Kathrin has discussed. So, Kathrin,
right.
What do you think?
Well, yes and no. I mean, an N of 1 study is easier to get the IND for, right? The conditions are not exactly the same as if you are planning to run a larger clinical trial. But what you can expect in terms of outcome and information is also, unfortunately, very minimal. I think the best solution is really to try and collect the natural history of the patients that you are planning to treat. Even if it's sometimes you don't have a choice because it's a really rare disease, but even in a more frequent disease, maybe really run a very tailored natural history study, which again costs money, but to really try and understand the patients that you are enrolling or thinking of enrolling, so that you know how their disease was progressing before, and maybe get specimens so you can do biomarker analysis before and after treatment. But an N of 1 really, there are other complications, also ethical complications, especially if you are developing a therapy that would be applicable for multiple patients. So is the patient that you're going to treat just the patient that can afford the treatment? That gets very complicated for nonprofit hospitals as well. So not always easier, I think. It really depends on what you're trying to do. If it's genome editing, that's a different story because it's tailored to the patient itself. And the hurdles are a little bit lower in terms of what you need to deliver to be able to get into the clinic for a single patient.
Yeah, thank you very much. I think this is it's an important topic. We actually, just for your information, we have the next webinar on May 14th is about this topic, the N of 1 gene therapies that will be presented by Terry Birovolakis from Elpida Therapeutics. So please join the next webinar. There will be more discussion about this particular question, which of course has a lot of challenges as well. So the next question relates to the SMA gene therapy. So Dr. Sahin is asking, long-term overexpression is a concern in SMA gene therapy, and one study in mice showed toxicity. What are your thoughts about this?
Right, that's a very good question. So it's not very well known, but the study that showed long-term toxicity did not use the same construct as the construct that is in the clinic. It's a different construct. Same promoter, but different construct. The promoter sequences are probably not the same again. That speaks exactly to what I said. If you wanted to do a comparison, you really need to be careful on how you're doing it. We haven't seen long-term toxicity in any of the wild-type mice that we treated. We haven't seen aggregates of SMA in the larger animals that we looked at. So it's an interesting study, and it will be really interesting, I think, to do further comparisons, but just be aware that they're not using the same construct.
Yeah, I agree. So I think the exact design of the construct, and including promoters and other elements that may enhance expression, are really crucial, and so it's important to compare the same constructs when you talk about toxicity, of course, and outcomes. So the next question is also about Zolgensma. Could intrathecal version of Zolgensma be beneficial for adult SMA patients too? So I think that relates to points raised by both speakers. So first of all, the timing, the reversibility, and then approved for one indication, and then can it be used for another phenotype and a different stage of the disease? So maybe both of you can provide a comment on this. Kathrin, if you want to go first.
Yeah, I can start. Yeah, I mean, that's certainly what we all would hope for. But it's difficult. We will have to evaluate it in the trials. I know the intrathecal trials are still ongoing and are still conducted by Novartis. I mean, technically, if the neurons are alive, we don't know how many neurons are there that can be treated. Increasing the SMN protein levels should be beneficial, but are we going to see somebody jump out of a wheelchair? You know, that would be fantastic, but it's also, I think, rather unlikely, because as the patients have been living with the disease for a long time, there is a lot of damage that happened, muscle wasting happened. So if the cells aren't there, or they're not connected to the muscle, or the muscle isn't there, you know, that we cannot change it. There are structural changes even to the whole body, like the bones, the spinal canal, and everything. So I always tend to be careful and say we could hope to either delay or slow down progression, or maybe even stop progression. But I wouldn't dare to spread, you know, the hope that we can reverse a disease until I see it in the patient.
Okay, thank you. Marta, do you want to comment on this particular question?
I think it's the same. I think we have to wait for data, but it's always benefit-risk decision. And I think given we know the safety aspects, and the patients who have been living with this condition already, they may not even accept those risks, but we have to wait for data.
Yeah, I mean, I think it means treating adults means higher dose, and also it's more difficult to show the benefit, I think, of the treatment because of the slower development of the disease. So it all becomes more challenging. The next question is also related to SMA. So SMA patients who already received Zolgensma may seek a second treatment, and this becomes more and more prevalent. What are your comments in this issue? I'm not sure. I think it relates to using probably the ASO, I mean, nusinersen
combination treatments.
Yeah. Yes.
Yeah, you know, it's an interesting development. Back in the day, a couple of years ago, you know, I've even had discussions with researchers saying, why should we even work on SMA? This is solved, you know. SMA has been treated or cured, etc., etc. But the fact is, AAV does not treat all the cells. It doesn't target microglia very well. Nobody knows how long it's going to last. And I think combination treatments could certainly have an additional impact. by either targeting cell types that aren't targeted by AAV, by either further increasing expression. As you said, the biodistribution is patchy of AAV, so you might get additional benefits. I think the problem is that all of these drugs, I mean, even the oral or the ASOs are expensive. So where do you, I think it becomes just a question of how do you manage the expenses versus the benefits. But I think any patient, if there is no treatment available, you'll be happy with any treatment you can get. But as you're feeling better, your wishes start to grow too, right? It's like, oh, I'm feeling a lot better, but if I could, have an additional benefit, I certainly would want to. And I think that's what we're seeing from my perspective.
Yeah, so I think there, in addition to the biology, we have other issues also coming in, including the financial and the expectations and possible long-term safety, all that. So I think it's not an easy issue, and of course, it's only for patients who have SMN2 copies. So the other question will remain in the SMA, even though gene therapy has been received in the pre-symptomatic stage, some patients still show some symptoms. Could this be related to the amount of motor neurons transduced? I don't know, maybe there is a question about also muscle related to the disease phenotype, but I think, of course, the percentage of motor neurons transduced will be directly related to the outcome. That's what I would predict. I don't know, Kathrin?
Yeah, I mean, I think there are a lot of variables, did the disease already affect the embryo before it was born, and to which extent? What are the disease modifiers? Some patients are just stronger even from start. Other patients maybe are already affected, but it doesn't really transmit into a visible defect. And then I do think there is variability in the viral vector uptake. And that is something that we haven't really, thought about too much. Other drugs, of course, nobody expects aspirin to work the same way in everybody, but for gene therapy, we kind of expect it. But there could be factors that limit the uptake in certain patients, and that would be, one of my guesses of why you see these differences in responses.
Yeah, but I think what's clear from the clinical trials and the real life experience is really very important, the timing of the treatment, just because of the development of the disease and the time period where the SMN is highly expressed and really important for motor neuron survival. So the other question relates to patients who are immune to AAV9. So I assume that's these are the patients who have neutralizing antibodies, currently cannot receive Zolgensma. Is there any way to destroy the existing antibodies to AAV9 so that this group of patients can also receive Zolgensma? So that relates to treating pre-existing immunity. Any thoughts on that?
Yeah, I think that there are a lot of companies and also researchers that are working on this. There are several approaches how to tackle this, which include, oftentimes apherases, trying to clean out some of the antibodies that are circulating, more aggressive immunosuppressant treatment, and even there are proteins that can digest antibodies in the blood. So there are several approaches that are currently tested. None of them is, very far in clinical trials or has even been advanced to clinical trials. But I think in the next 5 years, there will be a significant development there is what I hope.
Yeah, okay, very good. And there is another question about epithelioid neoplasm of the spinal cord in patients with SMA treated with Zolgensma. What could be the reason? Any thoughts?
No, actually, I don't think I have any answers to that. I also don't know, how many patients that has been seen in, because I think Zolgensma has been given to 3,700 patients at this point. Is this something that is unique to one patient or to a few? I would really, I don't have any information on that. Yeah, good question.
Yeah, we need to look it up. And the last question, I think I already asked this question to Marta. So what would be the EMA equivalent of the FDA investigator-initiated IND? So maybe we can repeat it once, just to before we close the
Yes,
questions and answer session. Yes.
Yeah, so there is no IND at all as such. So, and also you kind of have this two-level system in Europe that clinical trials are not done by EMA, they're done on national level. So if you have, if you work in a hospital anywhere in a member state, you can initiate that clinical trial after approvals from your ethics committee and local bodies. You don't have to have open IND to initiate clinical trials.
Okay, very good. That's clear. And maybe one follow-up question on that. So is there does EMA take any consideration of FDA evaluations, for example, for at the pre-IND or IND stage, or you work completely independently?
Yes,
in terms of decisions and approvals.
Yeah, so we have.
It's something that we always wonder about.
Yes, this is also, I get this question also for the guidelines. So in ideal world, it will be the same development across the whole world, but it's not happening. We do obviously exchange information. We do keep in touch on various fora, but our evaluations are independent. So you may end up with a different outcome for the same dossier in both regions, or different labels, or actually negative, positive. It's happening. We try to obviously explain why it happened, if it happened, but we don't strive for consistency.
Okay, great. Thank you very much. This was clear. And with this, we will close the webinar. I would like once more to thank both of you, Kathrin Meyer and Marta Kollb-Sielecka, for your very interesting presentations and the discussion we had, which was very insightful and informative. I would like to thank also all the participants who joined the webinar today. And please stay tuned for the following webinars. We have a number of webinars on various aspects of gene therapies that are important for different disorders and different stages of development. The next one will be May 14th. Please look at the EURO-NMD website for the entire program and to register. Thank you again, everybody, and have a nice evening.
Thank you, Marta. Thank you, Kathrin.
Thank you. Bye-bye.
Thank you very much. Thank you.
Bye. Bye.
Bye. Bye.